Let's bring in our next guest because it's going to kind of go in that same heartbeat of what we were just talking about, I think, a little bit. Alec deSimone, Associate Research Scientist, Yale University in the LEC lab, who Angela Leck we just had on. And Alec is connecting now as we speak, so exciting to watch the three dots turn into a check mark. Hey, how's it going, Alec?
Hey. Hi, great. How are you?
I am doing phenomenal. We just had a conversation with Angela Leck, and she knew you were coming on next because she was talking to us about some of the work that's being done there at the Yale University and some of the research that you guys are doing there. So, but first, maybe introduce yourself just a little bit. Tell us a little about what's your part in all of it there at Yale.
So I'm actually pretty new here at Yale. I was last year when I talked to you. I was still with the Wellstone Center at UMass Medical Center.
Yes.
So I was there for a good long time, for 6 or 7 years, and I had been working on a project there, largely taking the same type of approach that Angela just talked to you about, of kind of trying to look at DUX4 and figure out what about it, what happens after DUX4 comes on that results in the cells, it being toxic to the cells and the cells dying and resulting in the dystrophy. So we had both actually started in the Wellstone program around the same time, but we had taken kind of different technical approaches to ask a lot of the same questions. And since we were both part of the Wellstone program, we've been working together for quite some time and swapping data and bouncing ideas back and forth and things. And it became apparent actually in the last few years that even though we had started off studying things from very different angles, that we were kind of on the same molecular pathways or rails that she was talking about. So that led me to come down here so we could work together directly and really try to put our brains together and see if we could move things along faster together than we could at different institutions. It's been an odd year for me here because I kind of got here just as COVID was hitting.
Oh, geez.
Yeah, so I started here in February. So I was just settling in right when the kind of the lockdown happened and we had to sit at home for a while.
Man. So did that put in a little bit of a hitch in the giddy up there a little bit in the progress point? Or was it kind of difficult then to kind of get things going? Did it delay some of the work you were hoping to start achieving already in 2020?
Yeah, unfortunately it did. There were some projects I was working on at the time that I wanted to take with me and really start up immediately, and we had to put those on hold for a while. Fortunately, the shutdown wasn't too long, and we were able to get back to work in just a few weeks. And so things are in a much better place now. We lost some time, which is unfortunate, but I think we're back on track now.
Angela alluded to some of the work targeting existing drugs that have been approved by the FDA, but maybe they're out of patent, and maybe a little frustrating because there's not always financial backing behind those types of projects. Can you elaborate a little more on that work, maybe from your perspective?
I mean, mostly I just reiterate what she had said is, a lot of us as academics had taken this approach of it's much easier to get to a drug if it's already been through the approval process, because that can take years. And even once you've got a good candidate, it can take years for it to get through the FDA. So a lot of us have taken the approach of trying to repurpose existing drugs. But then what's wound up happening is, especially at some of the Wellstone meetings that we go to, where we meet with representatives from drug companies, and when we tell them that we're taking that approach, their attitude is typically, we don't want that. Because you can come to us with this drug and we can spend millions and millions of dollars doing clinical trials, but then there's nothing for us at the end, because then it just goes to generic, and then other companies will come in and sell it cheaper, and we did all the investment. So unfortunately, a lot of the time, drug companies aren't interested in that approach, which unfortunately slows things down.
Well, it seems like, when you say, drug companies and the representatives, it feels like that it's political, right? That if there's not money to be made, then why go that direction? Is you think that, as I mentioned or asked Angela the same kind of question, is there ever a point you see that we get past that to get something to us?
I would love for that to be the case. I don't know how much we can get past it, but we can massage it a bit. So one of the things that you can do is if you have a good like chemical formulations department, and you have a good candidate drug, you can tweak the drug into something new and generate new IP that way. And that gets the drug companies back interested again. It is kind of an extra step to get to, what is essentially a drug that we've had a very similar, alternative to already. But unfortunately, that's kind of just how it's played. You have to work within the system that we have.
When I was looking up some things, and I'm not a scientist, as you know, so you're trying to like identify a new compound. I was reading a P300. Am I kind of in this right world here? And using like a HAT protein, HAT. Can you explain a little of the science about that and how does it help within treatments or finding one?
I can a little bit. So that's actually not my work. I did write a brief blurb for that for the FSH Society about a year ago.
I grabbed it from, yeah.
Yeah, so that came from Michael Kaiba's group at the University of Minnesota.
Okay.
And the approach that they took with that is that, as you know, DUX4 is a transcription factor, which is our terminology for saying it's a gene that tells the cells what other genes need to be on and which ones need to be off. And a lot of the ways that transcription factors work is they work as complexes. So like DUX4 doesn't do anything by itself. It interacts with a number of other different proteins. And then as a team, they decide which genes need to be on and which genes need to be off. So a lot of the proteins that are transcription factors will interact with things that we call general factors or general transcription factors. And P300 is one of them. So it will kind of activate whatever gene it gets near to. And what DUX4 does is it binds to the gene and it binds to P300, so it brings them together. So then P300 will actually do a lot of the activating. So what Michael Kaiba's group has been working on is an inhibitor that blocks that interaction and stops P300 from activating the genes that DUX4 is directing it to.
Okay, gotcha. Thanks for explaining that. But you're right, I did pull that off of that paper that I saw, or that article I saw on FSHD Society. What are some of the current projects that you're personally working on within Yale University, currently that you can talk about?
So I can always talk in general terms.
Yeah.
The project that we've been mostly working on is to kind of follow up on some of the work that I did at UMass and some of the work that Angela was doing here before I came, and to identify additional compounds that look like they can help prevent DUX4 from being toxic, and that we can then move downstream and develop them into, hopefully develop them into therapeutics. So I work a lot with the cell models that we have in the lab, and what the cell models let us do is activate DUX4 when we want to, in kind of a robust and uniform way. And then we can see how that affects the cells. So we can identify drugs based on the literature that are known to work within the pathways that we're already studying. And we can add those to our cell culture models, and we can see if we then activate DUX4, do the cells still die? Do they take longer to die? Do they look healthy otherwise? Can they still behave the way cells are supposed to behave? And then kind of from that, we can then move those into other models that are kind of more physiologically relevant. And then if they all look good, then we can move them into animal models that Angela alluded to earlier. Yeah. So then we can either inject them into a mouse or feed them to a mouse, and then see, does the mouse behave better? Do their muscles fare better when they have DUX4 expressed than they would otherwise?
And so you're not quite at the mouse model level, right? Yet.
Myself, I'm not working at the mouse model. We do have one drug that we're working in the mouse model with right now.
Okay.
And it's looking promising, so hopefully we'll be able to move that further down the line. I'm mostly working in the cell culture models now to identify other, alternate chemicals
Other
that we can
ones.
put in the mouse, and hopefully they'll be even better than what we have.
Okay. So if you're working on the cell level, I'm starting to get this. Of all the researchers I've had on today, Alec, I'm starting to get the flow more and more. So if you're working at the cell level and I see it like a assembly line, so to speak, in my primitive brain. And if you're working the cell level and then you pass it down to those that will work at on the next level, what's coming down the pipeline for your area? Is there more what's the proper word? Possibilities, I guess, or drugs that you're working in with there or research?
Yeah, so there's several drugs that I'm working with now. Like Angela mentioned, some of them are actually really simple things that are supplements or even compounds that are known in foods that are known to work in the I don't remember if she gave the name of the HIF pathway that she found.
Mm.
So I'm working with those and hoping to pass those on to the mouse pretty soon.
Yeah.
The other thing that we're hoping to move forward to is that there's a whole library of very similar compounds that are also known to work in the same pathway, but with different properties. So we're hoping to really be able to get some funding to be able to expand that study into a much larger pool of compounds where we can really find one that has the properties we're looking for.
Oh. Okay. All right. I'm translating this as hope, Alec. I'm translating this as hope.
It is. Yes.
It is hope. That is great news. Yeah. And that's like, that's kind of why we're here, right? We're trying to help fund the hope a little bit. For you, how important then is this kind of community involvement? If it's fundraising events or I know you're working at the cell level. I'm not sure if you're getting, if it's cells created in the lab or if you're getting them from donation of people or not. But the question still remains: how important is it for community involvement, FSHD Society, etc., on days like this to help your work?
Oh, it's absolutely critical in a lot of different ways. One is one that you just mentioned. The cell models that we work with, a lot of them are direct donations that were given from patients that we can then keep going in the lab, and we can really get a lot of mileage out of them.
Yes.
Funding is also important. Like if I hadn't been funded by the FSH Society at UMass, I wouldn't still be able to be here to do the work now. And without further funding from not just the FSH Society, but Friends of FSH or the Carino Foundation, without the support of all these groups, we wouldn't be able to get the different chemicals that we want to test, and we wouldn't be able to maintain our mouse colonies. And it's just the work couldn't happen without the support of the FSHD Society and societies like it.
Yeah, it's promising work you're doing. I mean, the fact that you're helping people is a huge motivation. But you've worked on it, this kind of work from one place at UMass to now Yale. What kind of drives your motor, Alec? What kind of gets you going to attack FSHD to go after this work?
Yeah, so for me, like my journey to FSHD was really more happenstance than it was for a lot of the other scientists you talked to today. I came to it I had done my PhD working with transcription factors and transcription factor complexes, but I did it working in yeast. So the same yeast that make your bread rise or make your beer. We use those as a model system in my graduate lab. But as I moved my career forward, I wanted to do something that was more directly relevant to people's lives, which is not to say that work in yeast isn't. It's very indirect. So I wanted to be at a place where I could really see my work actually mattering to people sooner rather than later. And that took me to the FSHD community, because when I joined Charlie Emerson's lab at UMass, the reason that I wanted to join the lab was because they were working on a transcription factor that was directly relevant to a disease that was really affecting people. So I wanted to join for that reason. And that is a lot of what keeps me going, because, like Angela said earlier, the financial situation is not great. You never know. It's a very uncertain future. You don't make a lot of money doing it. But events like this really help keep me going. just seeing the supportive atmosphere that the community has. Like it's just a lot of people who need help and a lot of people who want to help, and just a mutual supportive, everyone's helping out everyone. And that really helps keep me going.
Well said, sir. Well said. And for you to be a part of it and let us know a little bit, peek inside, right? What's going on in your everyday work and working on our behalf. It's really well received, well appreciated, and glad you could join us again this year and talk a little bit about it. So we totally appreciate your time and taking time out of your Tuesday to join us today. We really appreciate it, Alec.
No, thank you. I appreciate you having me on.